Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you. Unlocking Life's Blueprint: The Power and Potential of DNA. Welcome to this episode of our podcast, where we dive deep into the molecule that holds the secrets of life itself, DNA. Deoxyribonucleic acid, or DNA, is at the core of every living organism, acting as the instruction manual that determines how organisms grow, develop, function, and reproduce. It's an elegant yet astonishingly complex molecule, and its discovery revolutionized biology. DNA is composed of two long strands coiled into a shape known as a double helix. Each strand is made up of building blocks called nucleotides, which consist of three parts: a sugar molecule, deoxyribose, a phosphate group, and a nitrogenous base. These bases come in four types: adenine, A, thymine, T, cytosine, C, and guanine, G. The sequence of these bases forms the genetic instructions for building and maintaining an organism, much like letters in a book form words and sentences. The bases pair in a specific way. Adenine always pairs with thymine, and cytosine always pairs with guanine. These base pairs are held together by hydrogen bonds, creating the rungs of the DNA ladder. One of DNA's most fascinating aspects is its ability to replicate itself. This process is critical for cell division and occurs with remarkable accuracy. During replication, the double helix unwinds, and each strand serves as a template for a new complementary strand. Enzymes like DNA polymerase facilitate this process by adding the correct nucleotides to the growing strand, ensuring that the genetic information is faithfully passed to daughter cells. The significance of DNA goes beyond just replication. It serves as the template for RNA in a process called transcription. RNA, in turn, directs the synthesis of proteins during translation. This flow of information, from DNA to RNA to protein, is known as the central dogma of molecular biology. Proteins, encoded by genes, perform a vast array of functions, from catalyzing chemical reactions to providing structural support and transmitting signals. But DNA isn't just about individual genes. Most of the human genome consists of non-coding DNA, once thought to be junk but now known to play crucial roles in regulation, structure, and possibly evolutionary processes. The epigenetic modifications on DNA, such as methylation, also influence how genes are expressed without altering the underlying sequence, adding another layer of complexity. In part 2 of this episode, we'll explore the history of DNA's discovery, how it shaped modern genetics, and the revolutionary technologies that emerged, like CRISPR and gene editing. Stay tuned as we unravel the story of this molecular marvel. Welcome back to part 2 of our deep dive into DNA. In this segment, we explore the discovery of DNA, the pivotal experiments that uncovered its role, and the groundbreaking technologies that have since revolutionized biology and medicine. The story of DNA's discovery begins in the 19th century. In 1869, Swiss biochemist Friedrich Miescher identified a substance he called nuclein in the nuclei of cells, which we now recognize as DNA. At the time, its significance was unknown. For decades, scientists assumed proteins, not DNA, were the carriers of genetic information due to their complexity and variety. This misconception persisted until a series of experiments overturned this view. One critical milestone was the 1928 Griffith experiment, which demonstrated the concept of transformation in bacteria. Frederick Griffith observed that a non-virulent strain of Streptococcus pneumoniae could become virulent when exposed to heat-killed virulent bacteria, suggesting a transforming principle. This principle was later identified as DNA in the landmark Avery-MacLeod-McCarty experiment of 1944, where Oswald Avery and his team conclusively showed that DNA, not protein, carried genetic information. The definitive proof came in 1952 with the Hershey-Chase experiment, which used radioactive labeling to show that DNA, not protein, was the genetic material in viruses. These experiments set the stage for one of the most iconic scientific discoveries, the elucidation of DNA's double helix structure by James Watson and Francis Crick in 1953. Aided by Rosalind Franklin's critical X-ray diffraction images. This discovery revealed how DNA could store information and replicate, fundamentally transforming biology. Fast forward to the modern era, DNA technologies have exploded in scope and impact. The Human Genome Project, completed in 2003, was a monumental effort to sequence the entire human genome, identifying over 20,000 genes and paving the way for precision medicine. Today, tools like CRISPR-Cas9 allow for precise editing of the DNA sequence, enabling scientists to correct genetic disorders, improve crops, and even explore de-extinction. Moreover, advancements in sequencing technology have reduced costs dramatically, making genomics accessible on an unprecedented scale. Fields like epigenetics and systems biology continue to reveal new layers of DNA regulation and interaction, showcasing its complexity. In the final segment, Part 3, we'll explore DNA's ethical implications, its potential for the future, and how it shapes humanity's understanding of life. Stay tuned! Welcome to the final part of our DNA exploration, where we discuss the ethical considerations, future implications, and the transformative potential of understanding and manipulating this vital molecule. As we've discussed, DNA is the fundamental code of life. It's what makes each organism unique and shapes the characteristics that define us as individuals. But with the increasing ability to manipulate DNA comes a set of profound ethical challenges. Technologies like CRISPR-Cas9 have made gene editing more precise and accessible than ever before, leading to groundbreaking medical advancements, such as the potential to cure genetic diseases like sickle cell anemia and cystic fibrosis. However, this power raises ethical questions about genetic modifications, particularly in humans. Should we edit the genes of embryos to prevent inherited diseases? What about creating designer babies, where specific traits could be selected? These questions force society to balance scientific innovation with moral responsibility. Another key issue is the potential for genetic privacy concerns. With the rise of personal genetic testing services like 23andMe and Ancestry.com, individuals can uncover not only their ancestry but also predispositions to certain diseases. While this information can empower people to make informed health decisions, it also opens the door for discrimination based on genetic information, whether by employers, insurance companies, or even governments. Ensuring that genetic data is protected while allowing people to benefit from the insights it provides is a complex issue that must be addressed as genomic technologies evolve. The future of DNA research promises even more exciting possibilities. We are on the verge of being able to tailor medicine to individual genomes, a practice known as personalized or precision medicine. In this field, doctors will be able to use genetic information to choose the most effective treatments, minimizing side effects and improving outcomes. Additionally, scientists are beginning to explore gene therapies to treat a variety of genetic disorders, potentially offering cures where none existed before. Another area that holds immense potential is synthetic biology, where researchers aim to create new, artificial life forms by designing synthetic genomes from scratch. This could lead to new ways of producing medicines, biofuels, and other valuable materials, as well as pushing the boundaries of what we consider possible in biology. At the same time, advances in DNA storage technology could revolutionize how we store and manage data, offering a density and stability far beyond current digital systems. Ultimately, the study and manipulation of DNA continue to reshape our understanding of life itself. From curing diseases to exploring the potential for life beyond Earth, the power of DNA is not just in what it tells us about the present, but also in how it will drive future discoveries. As we continue to unlock its secrets, it's crucial that we approach this power with a combination of curiosity, responsibility, and foresight, ensuring that the benefits of these advancements are shared equitably and safely. Thank you for joining us in this fascinating journey into the world of DNA, where science, ethics, and the future of life itself intersect. Stay tuned for more discussions on the cutting-edge topics that shape our world.